All right, here we are for our discussion, or at least the beginning of our discussion of enzymes. Now, there's a lot to unpack in this chapter, in this section. So be patient with yourself as you work through it. Now, the first question is, what does an enzyme do? This is going to be in contrast to our discussion of ligand binding proteins, that being hemoglobin and myoglobin. Enzymes catalyze thermodynamically favorable reactions, causing them to proceed at extraordinary rates. The most important part of that statement right there is, as I plug in my tablet, there we go, this word right here, catalyze. So they drive a reaction and cause a reaction to take place. And more than anything else, they cause these reactions to go at extremely rapid rates. Now, enzymes provide cells with the ability to exert kinetic control over thermodynamic potentiality. Basically, what that means is that an enzyme has the ability to kind of speed up a reaction or, in some instances, slow it down. Basically, they become the driver of a reaction. Now, living systems use enzymes to accelerate and control the rates of vitally important biochemical reactions, in that they are also the agents of metabolic function. So metabolism of sugar. So as I, drink this soft drink here, if there is any sugar in it, well, my body is going to control, or there are enzymes that are involved that are going to control this processing of this sugar. Now, there's, if you look it up now, you'll see there are a total of 7 major classes of enzymes. For this class, what we're going to do is we're going to focus in on a total of 6 of them. These 6 are the oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. Now, these major classes follow the IUPAC naming and numbering kind of categorization or organization scheme. This is a systematic classification of enzymes based on the results of the Enzyme Commission, which, if I remember the structure of it properly, the hierarchical structure, the IUPAC designated an Enzyme Commission to come up with a classification and categorization of every single different enzyme catalyzed reaction. Now, what I need you to do is I need you to know these these classes in sequence. So 1 are your oxidoreductases, 2 are your transferases, 3 are your hydrolases, 4 are your lyases, 5 are your isomerases, and 6 are your ligases. So if I said, what is EC number 5? I would expect you to say, oh, those are your isomerases. EC number 2 are your transferases. Those numbers and those names, link those 2 up in your mind. Now, here's a deeper dive into these 6 classes. Number 1 are your oxidoreductases. These are oxidation reduction reactions. So, in an oxidoreductase or an oxidoreductase enzyme is going to do a reaction where there are transfer of electrons, where there is oxidation and reduction taking place. Now, one example of that class is an enzyme known as lactate dehydrogenase. Lactate dehydrogenase, commonly known as LDH, what it does is it takes this molecule lactate and this other redox cofactor, NAD plus, I should correct that, and produces pyruvate and NADH. Now the key here, in the way that this ends up being an oxidoreductase, is this molecule right here, NAD plus, nicotinamide adenine dinucleotide. That has a positive charge to it in that form. What it's going to do is it's going to accept electrons in a hydride atom to make NADH. So NAD plus accepts electrons from lactate to make NADH. And the opposite is also true. Lactate is produced when pyruvate accepts, or gives up electrons to NADH. So, accepts electrons from NADH. So an oxidoreductase, typically what you're going to see in an oxidoreductase is you're going to see NAD plus. I'm going to say NAD plus slash NADH. NADH. Another, and I'm going to label this as redox cofactors. You might also see FAD plus and FADH2. You might also see NADP plus and NADPH. Then, in addition to all of that, you might see something like, well, Cu2 plus and Cu plus, or Cu2 plus and Cu. So essentially, a species is going to accept or donate electrons in an oxidoreductase reaction. So that is one of the hallmarks of an oxidoreductase, the transfer of electrons. All of those are examples of redox cofactors that you might stumble upon or might encounter whenever you're talking about an oxidoreductase. Now, with that in mind, I want to elaborate on one other thing. If you look at this name, lactate dehydrogenase. Okay, so here's our name. Great. Lactate dehydrogenase falls into category 1 because it's an oxidoreductase. Now, lactate dehydrogenase is one example of an oxidoreductase. There are hundreds of other oxidoreductases. And so what you might have seen on that previous slide was that the previous slide had quite a bit of information. Now, these last 3 numbers right here, these are identifying lactate dehydrogenase specifically, and what they're doing is they're saying that this enzyme is involved in a couple of different processes as well. So I always think about this as any and every enzyme is going to have a 4-number scheme. In the case of an oxidoreductase, the first number is going to give you the overall class. Then there's going to be a second number, a third number, and a fourth number. These 4 numbers combined will identify a specific enzyme catalyzed reaction and a specific enzyme that catalyzes that enzyme catalyzed reaction. So anytime you see this sort of numbering scheme, think, okay, well, this is identifying everything. This is as detailed as possible. Now, every enzyme is going to have that. The front number is going to vary a little bit. The next group, or the next class of enzymes, are our transferases. I can't highlight red with red. Our transferases. Now, transferases, what they are going to do is they are going to transfer carbon, nitrogen, or phosphorus containing groups. That could be something like this. That group gets transferred. Or one that we've actually seen quite a bit of is a group like this being transferred, a phosphate containing group or a phosphate group. A phosphorus containing group, a phosphate is a great example of that. Now, with that in mind, one example enzyme that carries out this reaction is one of those enzymes from glycolysis, fructose 6-phosphate. Fructose 6-phosphate plus ATP is going to yield fructose 1,6-bisphosphate and adenosine diphosphate. So what we have here is ATP, ATP transferring a phosphate group onto fructose 6-phosphate to make fructose 1,6-bisphosphate and ADP. That's left behind. Now, phosphofructokinase, or PFK1, is the enzyme that catalyzes this reaction. It has a numbering scheme of, well, 2.x.x.x. This 7.1.11 is specific for phosphofructokinase. The next enzyme class are our hydrolases. What these are going to do is they are going to cleave bonds with the help of water. One example is urease. Urease is going to take the compound urea, water, or urea and water, and it is going to break that into CO2. Now, urease has a numbering scheme or a specific number that is 3. Okay, so that tells us that it's a hydrolase. Urease is 3.5.1.5. I don't expect you to know these numbers. What I expect you to know is that first digit. What's the significance of that number? 1, 2, 3. Now on to 4. Lyases. Lyases are a class of enzymes that cleave carbon-carbon, carbon-sulfur, and certain carbon-nitrogen bonds. Fructose 1,6-bisphosphate. If you remember that molecule from glycolysis, that was our doubly phosphorylated sugar that gets cleaved into an aldose and a ketose. Our aldose was glyceraldehyde 3-phosphate, and our ketose was dihydroxyacetone phosphate. Now, what I want you to notice about this is what's not necessarily lacking, but we've got one reactant going to 2 products. We have 2 phosphorylated products, and we have one doubly phosphorylated reactant. What makes lyases different, and this specific lyase different from our previous class, is we didn't have to have water involved in this reaction. An aldolase, the enzyme aldolase, has the numbering scheme 4.1.2.13. That 4 indicates that it's a lyase. And in this case, we're cleaving a carbon-carbon bond. I don't expect you to know the products or anything, or the specific structures of the products or anything like that from this. I just want you to get a sense of, hey, you've seen these before because we previously talked about glycolysis. Now, next up, our next group, which starts with a 5, are isomerases. One isomerase in particular, and so think about what an isomerase is going to do, or what an isomerase may do. It's going to take one form of the molecule and make an isomer of the same molecule. It's just going to kind of rearrange bonds. We're not breaking anything permanently, and we're not gaining or losing any atoms. If there is a break or if there is a gain or loss, it's not going to be a net gain or loss. 2 different isomerases. One isomerase or the first one from glycolysis is phosphoglucose isomerase. That has a numbering scheme of 5.3.1.9. That enzyme takes glucose 6-phosphate and produces fructose 6-phosphate. So it takes an aldose and rearranges it to a ketose. Okay, phosphoglycerate mutase, 5.4.2.1. 3-phosphoglycerate is rearranged to 2-phosphoglycerate. So we're just rearranging the atoms of this. That's all that an isomerase does. So when it comes to enzymes and like knowing their numbers, people always like to know isomerases because they're really just taking an atom or a molecule, and they're rearranging the atoms of that molecule. Now, the last one that we're going to talk about are the ligases. Ligases are responsible for bond formation between carbon and oxygen. Oopsies. Oxygen, sulfur, and nitrogen. So if you see something like this, that bond would be formed with the help of a ligase. Now, what's cool about ligases is that they are able to do something by making a bond. And they make a bond with the help of this molecule right here, ATP. Now, that's not the first time that we've seen ATP. We saw ATP when we were talking about transferases. But the difference between this ligase and a transferase is one thing that's missing from this reaction that I need to add in is adding plus inorganic phosphate. Now, that's very significant because that inorganic phosphate, if this were a transferase, that phosphate would end up on our product. But in this case, if you were to, just for the sake of discussion, like ATP breaks down into ADP and inorganic phosphate. So a reactant broke down into those two different products. So it's not like you have this phosphate group stuck onto some other molecule. Instead, you can just say, okay, well, I know where all the atoms that came from that ATP. Now, what this means is, or what this specific enzyme is going to do, is it's going to take pyruvate, ATP, and CO2, and it's going to add this CO2 onto pyruvate and make oxaloacetate. That oxaloacetate is going to be, well, it's one carbon longer than pyruvate. This is a carbon-carbon bond that's produced. All of this is because of this enzyme harnessing the energy of ATP hydrolysis. The enzyme pyruvate carboxylase does this. This has an EC number of 6.4.1.1. Now, there's a lot to unpack, and these are all complicated, but be patient with yourself. Think about what the reaction is doing or what these different reactions do, and you should be in good shape on this topic.